Hello, welcome to SnapRevise. Welcome to a session that I'm going to be running today on the light-independent reactions of photosynthesis. This is something that lots of people find quite tricky, so hopefully you are here as a Year 13 student, ready to make sure that you know absolutely everything that you should know about the light-independent reaction. I'm just having a look at my comments right now. I can't see any comments. This is the first time that I've seen so few of you commenting before. Very disappointing. Yes! Fatima, you have saved us. Fatima is here. Excellent. The aliens come in. Hello. Anisha, hello. Jaisal, hi. How are you doing? Right. So I am going to get you set up so you can see what I am seeing. And then we will get started. How do I do that? There you go. Here is my PowerPoint. Hopefully you can see something that looks vaguely like the other things you have seen. Yes, perfect. Right, let's get going then. Hi guys, hopefully you're all having a really nice day so far. Hopefully you found that your day's been quite fun. I don't know, maybe I'm ambitious. Just to introduce myself, if you haven't seen any of these videos before, I'm secretly hoping that lots of you are telling each other about these videos and you're starting to sort of spread the word a little bit. But I'm Oli V. I'm the head of biology here at SnapRevise. Basically, I've been a teacher for the last 4 years of my life, where I've taught quite a lot of A-level biology. So I've had a good few students going through, having been taught by myself. Before that, I did my degree in biology at the University of Southampton, and I've ended another degree at UCL in London. So hopefully I'm one of the best placed people at the moment to be helping you with all of this stuff. In case you are wondering what SnapRevise is, if this is your first time, hopefully, again, as I said, someone's told you about it and you might have some idea of this. Basically, the idea of these webinars is to basically make sure that you are recapping everything. So one of the most important things for A-level is that you are constantly recapping everything, you are seeing what you need to know, and you're sort of refamiliarizing yourself. So you're going from probably a bit of a dependent learner at GCSE to a totally independent learner, so that when you ultimately get to university, you guys are going to be in a really great position. I'm planning on going through all the key points of these topics that I'm doing, going over some exam technique. And of course, as you guys are currently doing, you can ask me questions throughout. So I'll try and keep an eye on this just so I can see who's got a few questions. Who loves biology? Love that already. Good start, guys. Oh, just so you know, before I forget to say, because I will forget to say things every now and then, for the first few weeks, you've got these webinars for free. Hooray. Unfortunately, in about 2 or so weeks' time, these won't be free. So on the 16th of October, we're going properly live with SnapRevise 2.0. And if you are willing, if you are enjoying it, then you can sort of pay us a bit of money and you can carry on getting these great videos, hopefully. Hopefully you find it great. However, we do have some free accounts to give out. We have one free account to give out, and we also have a big old discount. So for any of you guys who are wanting to get SnapRevise 2.0, if you want a very, very easy way to get these webinars, all you have to do is follow us on Instagram, and you have to basically share a picture of yourself having a fantastic time or having a screenshot of you guys watching one of these videos. Someone's going to randomly get picked out, and we'll give you a free account for SnapRevise 2.0. So you'll get more of this. Let's have a look. Someone said biology is the hardest subject. Hopefully not for too much longer. I also have a coupon. If you manage to stay right till the end, and because those people ask me every time how long these sessions are, I'm aiming for this to be about an hour. It might be just a bit below, it might be just a bit over, but roughly an hour is what I'm aiming for. If you can stay right till the end, we've got a big discount code for you, which will be 40% off of SnapRevise. I don't know what else I can really say about it. Yeah, stay tuned. You can use that voucher right up until midnight tonight, I believe. Okay, right. What I like to do then is, and again, you've probably seen this before, I like to find out what you guys actually know about photosynthesis. So those of you that saw my session last week, where we did the light-dependent reactions, hopefully these questions will be something which you are familiar with. So the first question, if I start down here, I always start at the bottom and work my way up. What is the symbol equation for photosynthesis? Let's have a look. Who can tell me what is the symbol equation for photosynthesis? Nice basic GCSE start. What do we know? Oh, you guys have been slow today. No? No one knows? I'm going to have to write it down in a second if no one can write it. Ah. Very disappointing. Okay, so the simple equation that I would expect you guys to know for photosynthesis is we have carbon dioxide. So we have CO2. I think I'm going to write just this one. We have carbon dioxide. Yes, someone has done it for me. Carbon dioxide plus H2O. When we react those two things, we make oxygen, which is the thing that everyone remembers because they remember the plants give us oxygen, plus glucose, which is C6H12O6. Guys, you've managed to come to life. Thank you very, very much, Jaisal. If I'm saying your name right, apologies if not. The aliens come in, excellent. Hyber, excellent. Be careful, those of you who are saying equals in your little equation there. I prefer an arrow. I know that's probably quite difficult to do when you're on a laptop or your phone or something, but I prefer that. Excellent. Some of you are balancing this as well. If you're balancing it, you've just got to remember the number of the beast. 6, 6, 6. Okay. You never have to really worry about balancing equations in biology, which is nice. This one, you just need to remember 6, 6, 6. I don't really think I can remember many other ones apart from maybe a couple of ATP ones that you need to really worry about. Yeah, fair enough. Don't worry if you couldn't do the arrow. That's absolutely fine. Right. Oh, Fatima, I love that you're trying to give yourself a head start with this. Excellent. So can anyone tell me about the organelle? So the organelle, the little organ, that literally means like L means little, organ means organ, little organ where photosynthesis takes place. Lovely. Lovely. I'm not going to say the answer just yet. I just want to see you say it. Bonus points. Can anyone tell me what part of the organelle has the first bit happening? Where does the light dependent part of photosynthesis happen? Can anyone tell me? So loads of you are saying chloroplast to get that first answer right. And you're saying that it contains chlorophyll. Ooh. So the part which is involved in the light dependent stage is the thylakoids. Yeah, it's the grana, it's the thylakoids. So describe the organelle where photosynthesis takes place. It is definitely the chloroplast. It's definitely happening in chlorophyll, which is a pigment. And yeah, I would say it's the thylakoid membrane or the thylakoids or the granum where the light dependent stage happens. Right, next question. Who can tell me this? So describe how carbon dioxide enters a plant. If you remember this, I'll be impressed. Who remembers how carbon dioxide can get in? Yes, someone has said it. I'm going to wait for a few more comments. Yep, lovely. So a couple of you are saying it. Lovely, lovely. I'm loving that you're even saying how it's happening. Perfect. So yeah, so how does carbon dioxide enter? So leaves, if you imagine my hand being a leaf, they have tiny little holes in the bottom. So very, very small holes made up of two cells called guard cells. The holes are called stomata, or as some of you are saying, stoma, if you're talking about more than one. Okay, stomata are little holes. They open and close that allow carbon dioxide in. They allow oxygen out. Those of you saying it happens by diffusion, lovely, even better that you're remembering that. Okay, right. This is all GCSE stuff in my mind. What about this next one? This is AS stuff, or back when I was at school, that's what we called it. That's like year 12 stuff. Who can tell me what a condensation reaction is? Who remembers what a condensation reaction is? I'll give you a couple of seconds. Okay. Ooh, I'm seeing people are saying two different things. Someone's saying the addition of water, someone's saying releasing water. Okay, condensation. Think about what condensation is. Think about what condensation is. Think about what hydrolysis is. Think about the term in hydrolysis, or like the last bit of that word means. That will probably help. Right, so condensation is something where you produce some water. So condensation will produce water, and it basically binds two things together. So a condensation reaction is where you synthesize something, you bind two things together, and that will make some water or make a bit of condensation. That's how I tend to think about one. Well done. Excellent, guys. Really, really good. Hydrolysis, then, the opposite, hydrolysis. This is the word that you cannot interchange with breakdown. If you do that in your exam, they're going to frown at you a lot. I'll be very upset. So hydrolysis. Lovely, lots of you are saying this. So hydrolysis splits a molecule using water, splitting of molecules by adding water, add water, breaking bond, breaks bond, addition of water, uses water. Nice. Yeah, good. So colloquially, I would say it is basically breaking something down. However, hydrolysis, we've got to know that it's when you basically have one big thing and you make it a little bit smaller. Breakdown is fine for like talking now, but I wouldn't ever say anything like that. Make sure you say hydrolysis. Okay, last thing then. Who can remember this? What are the products? Key question. What are the products of the light-dependent reaction? After we have finished the first stage of photosynthesis, what are we left with? So some people saying some different things. I like that you guys are, this is amazing. So everyone is saying ATP, which is spectacular. So there is some ATP made, brilliant. Some of you have remembered that we have some oxygen being made. So we split our water down in photolysis. Photolysis breaks down water, which leaves us with some oxygen. Lovely. And then lots of you are saying something like this. You're saying NADPH. So about half of you have said NADPH, and about half of you have said reduced NADP. Okay, I would try and get in the habit of saying the latter. I would try and get in the habit of saying reduced NADP, and then as soon as you've said it once in an exam, if you put in brackets after NADPH, that's just showing your examiner that whenever you say whenever you say NADPH, you actually mean reduced NADP. I think it's pretty much accepted that NADPH is reduced NADP. However, you don't want to have a horrible examiner who's just being harsh for no reason. Okay, so I'd get in the habit of saying reduced NADP, but that does mean NADPH. For AQA, I've only seen AQA examiners doing that. Okay, that is probably because I've worked more with them than anyone else, but yes, all of this is going to cover all the exam boards as well, Diana, or Diana, however you say your name, just so you know. Lovely, right, so what we're going to do then, and the eagle-eyed amongst you will notice that I have slightly changed one of my learning objectives if you've printed out a little booklet. I want you by the end of today or by the end of this evening to basically be able to understand the Calvin cycle. Okay, the Calvin cycle is just another name for the light independent reactions. Okay, and it's light independent reactions. There you go, said it. I need you to understand the limiting factors of photosynthesis. Okay, so I thought these two things go together quite nicely, and I think you get questions that sort of cover all of this. And finally, I need you to understand basically how to answer photosynthesis questions. So if you have the other booklet that you've downloaded, I have slightly changed this because I would rather go a little slower through the latter bit so I don't have to squish it all into one thing. So I've decided to do a few more exam questions and take out one last little learning thing. Just so you know, this live stream will be roughly an hour. Okay, so about 40 minutes from now-ish, or 50 minutes from now, just so you know. Okay, so things that I won't cover. Cyclic photophosphorylation. This was something that I spoke about basically at the end of my last session. I'm not going to talk about that because it's more to do with the light dependent reaction, and it's only really covered by one of the exam boards, so I'm not really going to worry about that too much. I'm also going to carry on not really worrying about the other photosystems or the other pigments. So I'm not going to talk about xanthophyll, I'm not going to talk about carotene or any of that. I'm just going to leave this sort of in the way that will hopefully be beneficial for the vast majority of you. If you are wanting to cover these things, I've noticed, you might have seen this as well, if you go onto our YouTube website, if you type in SnapRevise and look at our videos, you'll see literally hundreds of videos of someone just going through the main bits, right? They don't have someone like me talking to you during it, but you can have a look at that, and hopefully that will cover these things. Okay, right, here are the spec points. Again, if you are revising, you need to be looking at these. You need to be checking out if you can do these things. You literally need to work for it and be like, oh, can I talk about the fixation of carbon dioxide? Yes, lovely, I'll move on. Can I talk about triphosphate? Lovely, I'll move on. Can I talk about these other things? Perfect. That's what you need to do. I'm not going to talk about them any more than that. These are for your own reference. Okay, they are the Edexcel ones. Hopefully you know what spec you're doing, either Edexcel A, B, AQA or OCR. How are we looking? Oh, the person who's just said that they didn't see last week's one, if you go onto YouTube and you go onto our videos, you will see one where it says the light dependent reactions. Okay, so you can watch that as many times as you like, go over it as many times as you like. But yeah, that'll be on YouTube for the foreseeable future. I think possibly forever until YouTube doesn't exist anymore. So lucky you. Right, so part one, we are going to look at the Calvin cycle. Okay, so this is going to get a little bit complicated, and I'm going to try and keep this hopefully straightforward. So in terms of what the Calvin cycle is it's firstly really important to understand sort of where it's going on. Okay, and it's really important to realize a bit of a contradiction in the biology here. So the light independent reaction basically uses all of the stuff that we made in the light dependent reaction, and it's sort of a bit stupid that we therefore sort of refer to it as the light independent reaction. So to sort of get my point across, like if you are gonna refer to yourself as not being something, so independent of light, if you rely on all the products from light, it seems a little bit hypocritical to me. So I think it's quite important that we realize that the light independent reaction is a little bit dependent on light, to be totally honest. It needs the products of light, otherwise it wouldn't really be happening. Hopefully that sort of makes sense. Like it just seems a bit of a contradiction to me, but we'll carry on, we'll ignore that. Okay. In terms of where everything happens, it's important as well that you understand where stuff is going down. So who can tell me what was going on in the thylakoid? Can anyone tell me what was going on in the thylakoid? And yes, Fatima agreed, silly people. What is the stage that happens in the thylakoid? Does anyone know? Yeah, exactly. So we are absorbing light, someone has just said, thank you very much. So in the thylakoid, this is where the light dependent reaction is happening. It's the light dependent reaction is happening in the thylakoid. Okay. In terms of the inner membrane, so in terms of this space, which is sort of between the two membranes, so the inner membrane space, again, this is the light dependent stage. Okay. So both of these two parts here, and this is a really, really standard A-level biology question. You'll have something which will say something along the lines of or describe specifically at which part of a chloroplast the light dependent stage occurs. Right? I see that question all the time. So they want you to be able to say the thylakoid and the inner membrane and stuff like that. Okay. In terms of where the light independent reaction is happening, that is happening in the stroma. Okay, so this is where the light independent reaction is happening. Okay, so the light independent section is happening in the stroma. I think if you saw my last session, I was basically banging on at the end about how you make ATP and you make NADPH or reduced NADP. You basically make all this stuff in the light dependent section, and then at the very end, it sort of kicks it all into the stroma. So it gets rid of all of it and chucks it right into the stroma just so it can be useful later on. Okay, lovely. So you guys are kind of, I think you guys are on board with this. In terms of this reaction then, I think it's also really important that you understand where each of these things is sort of happening. So in terms of CO2, but let's not start there, let's start with H2O. What section of the lights, what section of photosynthesis is H2O being used? So I'll get to you in a second, the person who's just asked me about diffusion. So H2O, whereabouts is it being used? Is it being used in the light dependent stage or the light independent stage? Who can tell me? Lots of you are saying photolysis, lovely. When is that happening? Light dependent or light independent? Lovely. So H2O is being used in the light dependent stage. Okay. CO2, on the other hand, if you've watched my previous video and if you've done this at school, you'll see that CO2 is actually never used at this stage. CO2 is never really used at this stage. CO2 only will like diffuse in, start being important in photosynthesis by the time that we get to the light independent. I also apologize for my handwriting. I've just seen how horrifically I've just said, oh no, that's an arrow. Apparently this guy here is meant to be an arrow. Possibly have just made it worse as well. So sorry, CO2 is being used in the light independent stage. Okay, so CO2 is being used then. In terms of when these different things are made, who can tell me when oxygen is made? Who remembers when oxygen is made? Who's going to be the first? No? Photolysis, yes. Yes, yes, lovely, perfect. The aliens are coming, watch out. Beautiful spelling there. I love that you're going for speed as opposed to accuracy. I think I'm on the same wavelength as you. So yeah, oxygen is being made in the light dependent. I'm just going to write dependent as opposed to light dependent. So oxygen is being made in the light dependent. Someone, I think it was Fatima, said that it is a waste gas. Yeah, lovely. Oxygen serves no purpose really to the plants in photosynthesis. Some of it can be used for respiration in the same way it can for us, for aerobic respiration, but it doesn't really serve much purpose. Most of it just diffuses out of the stomata. So this carbon dioxide diffuses in and just diffuses to the stroma. Oxygen will diffuse out. Okay, glucose, the final bit, the most important bit in terms of us. This is made in the light independent stage. Right, that is looking already quite complicated, okay? But I do think it's important that you have a good understanding of sort of what's being made where, what's going on where, because if you have an idea of that, I think it makes everything else sort of piece together a little bit easier, okay? Water is a byproduct. In terms of respiration, it is. In terms of photosynthesis, it is not. The aliens are coming. I'm glad that you're wanting to be the first. Love it. Sorry, yeah, and just to get to this point here, so the product of the light-dependent reaction, just to say it again, I have said it already, this is ATP and it's NADPH, or as I said, refer to it first as reduced NADP. Okay, yes, GALP I'm going to get to in a second, those of you who are saying GALP, which looks like a weird sort of exclamation of someone who's just been trodden on. GALP I'll get to in a second. Okay, so this diagram here is showing you the Calvin cycle. Okay, and it looks a bit complicated, and I'll sort of talk you through what's going on first. So the Calvin cycle is a way of producing glucose. Okay, so this little cycle going on here, I don't know why I'm going to draw a little circle in the middle. Maybe I'll draw a little smiley face. There you go. The Calvin cycle is a way of producing glucose. Okay, this molecule down here, this 6-carbon molecule, this is basically, I'm not going to call it glucose on purpose. I'm going to call it a hexose sugar. Okay, that includes glucose, but it could include some other things as well. It could include something like, can't think of anything. Could include something like, can anyone help me out? I'm not thinking sucrose, or am I thinking sucrose? No, something like fructose. There you go. Fructose would be another example of something you can make. Okay, so the Calvin cycle is a means of making hexose sugars, and that happens down here. So for those of you who are confused why this says TP, I can't remember which board it is. I forget which way around it is, but basically all of these little initials, so RUBP, that one stands for ribulose bisphosphate. Okay, GP stands for, I think it's glycerate-free phosphate, and TP has, I think it's glyceraldehyde. I can't remember this, and you guys don't need to know this, don't worry. It basically has different ways of writing it as like 2 initials. So there's one example that refers to TP as GALP. So just so we're all aware of this, one of them says GALP. Yeah, AQA certainly says TP. When I've taught this probably for 3 out of 4 years to AQA, so I know for a fact they call it TP, but I know there's at least one of them. I can't remember if at the previous school I taught at they called it GALP. It doesn't make a difference. It's just they choose to call one thing one and one thing another. There you go. GALP is Excel. Lovely. Thank you, Ewan. Okay, so this looks, again, really mentally hard, but it's really not too tricky. Essentially what's going on is I've got a molecule called RUBP, and I always try to remember this as, it's kind of like it's asking you a question. It's like saying like, are you BP? I don't know. Am I BP? I just remember it like that. It sounds stupid, but that's the way I've remembered it. So RUBP. So RUBP is just a molecule made of 5 carbons. So these little kind of purple things that you can see, which I'm sort of slightly colouring in, these are meant to be carbon atoms. I hope someone liked that. So these little guys here, these are carbon atoms. Okay. Carbon dioxide, as you know, CO2 is a nice basic little compound. It is made of just 1 carbon. So I've got a 5 carbon compound joining with a 1 carbon compound, and because you guys can do maths, that ends up making something which has 6 carbons in it again. So 5 plus 1 is 6. That's straightforward. Okay, this big molecule here, which we don't ever have to worry about its name in any of the boards, this very, very quickly breaks down to form these little 3 carbon molecules. Okay, it happens to form 2 because something which has 6 carbons in basically breaks in half to form something with 3 carbons in. Okay, these 3 carbon things, they go through a bit of a process and they turn into a slightly different 3 carbon compound. Okay, and then you're going to have to do some maths in a bit, and I don't mean very hard maths. These two 3-carbon compounds can magically turn back into a 5-carbon compound. Okay, so there are some different steps going on. Someone has said a good question a second ago. Maybe they haven't. Oh, good. Yeah, someone has said this. So 1000 subs, no videos has said that where it says ATP here and NADPH here, it actually means 2 of them. So it actually means 2 ATPs and it actually means 2 NADPHs. Okay, this is because each of these little GP molecules needs a bit of ATP to get to turn into TP. Okay, so there'll be one ATP that joins with this GP, and there'll be another ATP that joins to this one. Okay, I'll get to that in a second. So I think the first place to start, and I'm going to break this down into a few different sections, and then we're going to go over it as a whole. I think the first really important thing is just to know what's going on in this stage here. So I'm just going to talk about this stage here. Okay, so I've said at the top that the Calvin cycle uses protons from NADPH and the energy from ATP to fix carbon dioxide and convert it into glucose. Okay, the first stage, oh, someone's asking a question as well. Where does the ADP go on the left? It will just diffuse off probably back to the light dependent stage. So it'll probably just go back to the thylakoids and become ATP again. Lovely. Someone's just said something I wanted to hear. So the very, very first stage which is going on is RUBP and CO2 react with one another. And that reaction is called carbon fixation. So the first thing that happens is carbon fixation. Okay, and carbon fixation is it's a reaction that can happen by itself. However, it needs to use an enzyme to make it quick enough for plants to be able to do photosynthesis efficiently. Okay, the enzyme that does this is called Rubisco. Okay, so Rubisco, I'm going to say catalyzes, because that's how you need to talk about enzymes, catalyzes this reaction. Okay, so we end up with RUBP, ribulose bisphosphate, reacting with carbon dioxide to make this 6-carbon compound, which is over here. Okay, that is called carbon fixation. Shouldn't say ADP plus inorganic phosphate. Good question, Fatima. No, the inorganic phosphate has been added to the molecule, so it doesn't have that as well. Okay, just so we're aware, I think this is a really interesting, I say really interesting, you're going to realize quite quickly if you watch lots of these videos that what I find interesting, most people find actually extremely boring. But I'm a biologist and therefore I can find this interesting and not be too embarrassed. I find it quite interesting that Rubisco makes up 30% of the protein in a leaf, right? So what, I hear you cry. That means that it is the most prominent enzyme on the planet Earth. So there is more Rubisco on planet Earth than any other enzyme. I think that's quite cool. Okay, so there is quite a lot of this stuff in existence, and really irritatingly, it's actually really inefficient. It's not very good at doing its job. Just as a bit of context, though, as we are living in a society, as I'm sure you are aware, that is pumping more and more carbon dioxide into the atmosphere, and I'm sure lots of you saw Greta Thunberg's speech the other day, which was amazing. You probably are aware that we're not very good at doing anything about carbon dioxide, and we are trying to recreate what Rubisco does in terms of in an engineering kind of way to get rid of carbon dioxide, but we're rubbish at it. So we've tried to make our own little sort of artificial Rubisco, but we're even worse at it than plants are. But yeah, Rubisco is very, very important. Like if we didn't have this enzyme, then this planet would be considerably hotter and probably wouldn't have any humans on it at all. Yeah, I think that's quite cool. I'm glad you guys are interested in that. So Rubisco, really, really important enzyme. Okay, hopefully just with this little bit here, hopefully we're okay. So I'm going to put a big old 1 here to say that this bit here is called carbon fixation. Okay, there's nothing really more to this. You just need to know this first step is called carbon fixation. Okay, so the second bit that's going down is going to sort of talk about this stage here coming round to about there. Okay, so we're going to talk about this stage here, and I'm going to sort of talk about it in 2 parts, as you can probably tell because I've written 2 and 3 down. Okay, so the first thing that happens, as soon as Rubisco has catalyzed this reaction of ribulose bisphosphate and carbon dioxide. Oh, one other fact, Rubisco is also one of the coolest enzyme names as well. I think it's called ribulose bisphosphate carboxygenase oxygenase. I think it's all in one word as well, so that's another cool fact about Rubisco, just for future reference. I got distracted. The next stage that happens then, after I've done my carbon fixation, and you need to talk about it in an exam that way, is my big old 6 carbon molecule here very, very quickly turns into 2 free carbon molecules. So my big, person's asked me how to spell it, Google it. This big old molecule that I've made is very, very unstable. So I would say that over here, you want to say a 6 carbon molecule will quickly, or you could say spontaneously if you wanted to, a 6 carbon molecule will quickly, you could say break down or hydrolyze. I'm going to say break down for now, just to be nice and straightforward. We'll break down, forming 2 molecules, 2 times GP. Okay, so that's the first little thing that will happen. So my 6 carbon molecule will quickly break down, forming a 2 molecule, a 3 carbon GP molecule, but there are 2 of them. Okay, this all happens very, very, very quickly. So this molecule here, I don't really know what the point is of you guys learning about it, and I've certainly seen in CGP's books that they tend to just ignore this quite a lot of the time. Right, it exists for such a short time that most people just ignore its existence. But I guess it probably makes sense for us because we started with a 5 carbon thing. It's added to something which has 1 carbon, and that makes something with 6 carbons. If we just said it formed 2 3 carbon things, I reckon people would get confused and give up. Do you have to write the names of the full sugars, or can you use shortened versions? Shortened versions are absolutely fine here. Okay. The next important thing then, so this is the second stage. So if we had carbon fixing was happening somewhere up here. Right, the next stage here, I would refer to as reduction. Okay, reduction is really important. Who can tell me what I mean by reduction? Let's get you guys thinking again and typing and having to actually use your brains for a sec as opposed to just listening. Who can tell me what I mean by reduction? Anyone? Oil rig. Excellent. Oxidation is loss. Reduction is gain. In terms of oil rig, you're totally correct. What is it talking about the loss of? I think lots of you are saying it. So oxidation is the loss of electrons. Reduction is gain. Reduction also means something a bit different in biology. Reduction, well, it's not different. It's just a similar way of saying it. Reduction also means gaining. Oxidation is loss. Reduction is gain. Reduction is where you basically gain hydrogen. Okay. In fact, I'm just going to write hydrogen. I'm not going to write it as an ion. Okay, because it's not an ion. So reduction is where you gain hydrogen. Okay, oxidation, easy to remember, gain oxygen. Reduction is the opposite, or a bit different. Reduction is gaining hydrogen. So what's going on over here then is my GP molecules are going to gain this H. Okay, we're going to gain this H each. Okay, so over here, I would say my 2 GP molecules, can't spell molecule, C-U-L-E, are reduced by NADPH. If it's been reduced, so if GP has been reduced, that means that my NADPH has been oxidized. Okay, you might be able to scrape a little mark as well if you can say that. So because NADP has reduced something, it itself has been oxidized. Okay, as soon as my NADPH has lost its hydrogen, it's going to go right back to the thylakoids so it can gain some again. So I can't remember if anyone remembers me saying this, but you basically have this system of NADP constantly moving back and forth. So it's like a little coenzyme is a term you use. So it basically jumps from the thylakoids to the stroma, back to the thylakoids, back to the stroma. And it's basically this repeated cycle which will carry on going. Okay, so my 2 GP molecules are reduced. Okay, this section I would call just reduction. So the first section, carbon fixing, second section is reduction. Okay, so each of my little GP molecules is going to have some hydrogen added to it. There you go, there's a nice little hydrogen added to it. Okay, if I add ATP to something, does anyone know what it's called? So it's reduced if you add hydrogen to it. If you add a phosphate from ATP, does anyone know what that's called? Lots of you are asking questions, so hang on, I'll go back and answer them in 1 second. Yes, it does move by diffusion, Matt Person. That's a nice quick one. What's it called if you add ATP to something? Lovely, lots of you are saying it. So 2 GP molecules are reduced by NADP. Let's say they are also, and then someone said it, phosphorylated. Okay, phosphorylated means it's just having phosphorus added to it, inorganic phosphorus, or phosphates even. So it's having inorganic phosphates added to it. Okay, if it has carbon added to it, that's called carboxylation as well, just that's something you actually see. So go on, let's have a look at your questions, because I think some of you are getting confused. Does NADPH mean reduced NADP? Yes. Where does stage 3 happen? Stage 3 happens up here. You're getting ahead of yourself. Could you explain carbon fixing again? Oxidized, phosphorylated, lovely. Right, I'll go through this again very quickly for this bit. So carbon fixing is just where you add CO2 to ribulose bisphosphate, RUBP, right? And that's catalyzed by Rubisco. That's stage 1. Stage 2, reduction. The hydrogen from NADPH is added to GP. Okay, so the hydrogen from NADPH, reduced NADP, is added to GP. Okay, when you do that and add a little phosphate group to each one as well, there's a little phosphate group. There's a little phosphate group. When you do that, it basically turns into this new molecule called triose phosphate, TP, or you can call it GALP if you are doing Edexcel. So carboxylation and carbon fixing are the same. Not quite. They are a little bit different here because this is CO2 as opposed to just carbon. Okay, they are basically the same, but carboxylation and carbon fixing are pretty much the same, slightly different. I think for the Calvin cycle, you just need to be able to say that it's carbon fixing. First time seeing this chapter, must be really behind. Sorry. Where does the phosphate come from? So ATP has been made during the light-dependent stage. So if you remember, you have a whole load of hydrogens moving through an ATP synthase, which is constantly spinning around. And when it does that, when hydrogen moves through this enzyme, it makes ATP. Okay, that's where this ATP is coming from. Is this for AQA? This is for everything. What happens to the ATP? So the ATP will basically be hydrolyzed, breaks off one of its phosphate groups, donates it to GP, and then ADP, which is left. So ATP is made with 3 phosphates, ADP is made with 2. The ADP will just diffuse back to the thylakoids, where it will go and have some more inorganic phosphate added to it. Okay, so the ATP will donate its phosphate, and then it will just go back to where it came from, essentially. Hopefully that's cleared up. I'll answer a few more questions in a second. Okay, right, final thing that happens then, and this stage is fairly straightforward. I don't think I really need to have these 2 stages. No, actually, I'm getting ahead of myself. So there are 2 things that happen. So with these TP molecules, they actually have 2 things that can happen. Okay, so these TP molecules can either be used to make my molecule down here, which is a hexose sugar, e.g. glucose. Or they can basically do what this stage is called, so I'm going to write it here, which is the regeneration, which sounds cool, regeneration of RUBP. Okay, so my TP or GALP, I'm going to try and use both. My TP or my GALP will either be turned into hexose sugars, like glucose, or they'll be used to remake my RUBP. And I'll get into, those of you who I know are going to ask me questions, and someone's just said it, about how many times this needs to go around this cycle. I'll get into that in a second. Okay, but just to keep it simple for now, we can say that my TP slash GALP, whatever exam board you're doing, just write that one down. Don't worry about the other one, is converted into hexose sugars. Or we can say it is regenerated, so used to regenerate RUBP. Okay, lots of you might be confused here. So the person who's just talking about 6 turns, 5 out of 6 things, yes, that person is totally right. I'll get to it in a second. So lots of people get confused by this. I think me being one of them when I did this. So I'll talk about that in a second. Can anyone tell me why this step here is so important? Why is it so important that I need to regenerate RUBP? Lots of my students in the past have said, why doesn't this GP just turn into TP and just make glucose? Why doesn't it just stop there? Can anyone tell me why we need to regenerate my RUBP? So the person who's just said, where does 4 and 5 happen? 4 is this bit here, that bit. 5 is this sort of section here, right? Mostly this is split into 3 sections. So we have our carbon fix in there, carbon fix in 1, number 2, reduction, number 3, regeneration of RUBP. Let's have a look. So the light dependent, sorry, so I'll start with you. Jaisal, am I saying that wrong? Am I saying that right? Please correct me if I'm not. He says to keep the cycle going, correct. Someone said to complete the cycle. Yes, I understand that we see the intrinsic need to complete the cycle, but to the plant, it doesn't matter. It doesn't care if the cycle is not completed. There needs to be a reason. So the light dependent reaction can occur. I kind of see what you're talking about there, yes. But not entirely. I guess it is supplying some ADP. So I actually forgot to mention that, didn't I? To turn TP back into RUBP, you need just 1 molecule of ATP. So you need to shove on a whole little phosphate group there. And to be fair to the person who said that, where did you go? I've lost you. So the light dependent can occur, Kitty. You are right, yeah. I do need to have a supply of ADP so that I can continue with my light dependent cycle. But that's not really the main reason. So the idea of regenerating RUBP is so that you can basically then make another molecule of GP. So you can then make another molecule of TP. So you can make another molecule of RUBP. So if you don't make that RUBP, if you didn't regenerate and you didn't make that, then you wouldn't have anything to react with carbon dioxide. So you wouldn't be able to make that. So you wouldn't be able to make that. So you'd just run out. Everything would stop. Okay. So if you don't regenerate RUBP, you can't make any more hexose sugars. So yes, it's a little messy that you have to wait a bit patiently to make your sugar, but you really need to make RUBP. Otherwise, this whole thing stops. Yay, Jaisal, I am pronouncing your name right. Thank you. Yeah, basically, the point of this is to make sure that glucose can be made. Could I recap the whole cycle after this? I'm so confused. Yes, I am planning to in a second. I've got one more little thing to confuse you before I do that, though. Right, in fact, no, this is the sort of whole thing. So let's go over the whole thing, right? So I put a blank slide in here just so I can sort of write everything down for you. So if I've got carbon dioxide coming in at the top, I've got a molecule here which has 5 little carbons, which is called RUBP. I hope YouTube that you are BP. I hope all of us are BP. But my little RUBP is going to move along. Carbon dioxide is going to react with it. And that makes my, I'm going to ignore the 6 carbon thing because it's there so temporarily, I'm just going to skip it. So this makes my 2 molecules of 3 carbon long GP. Okay. And then that GP turns into 2 molecules of 3 carbons long TP / GALP. And then that turns back into RUBP. Okay. So let's go through it in terms of the sections. So number 1, I'm going to write there. Number 2, I'm going to write here. Number 3, I'm going to write here. Okay. So number 1, what was the thing that happened during number 1? Let's make sure we've got this right. So number 1, the section is called what? What is section 1 called? Come on, guys. Yes, Nyasha. So number 1, I'm just going to write a little CF. Okay, section 1, CF. Hopefully, that's not nice. Hopefully you guys are really okay with this, okay? All that is happening is RUBP is reacting with carbon dioxide, and there's a little enzyme called Ribisco, which catalyzes that reaction. Okay, that's carbon fixing. That made this crazy little 6 carbon molecule, and then that broke down into my 2 GPs. Okay, Hopefully you guys are all happy as well that that's only got 1 carbon. So is everyone okay with these numbers so far? So I've got a 5 carbon molecule over here, a 1 carbon molecule here. That makes 2 3 carbon molecules. I know you can't all say yes at the same time, apart from Aisha. Cool. Right. So next stage then, to turn GP into TP/GALP, there's not a whole lot of difference between these two molecules. They're very, very similar. The only difference is that this one has had another molecule added to it, or 2 more molecules added to each one, right? It's had NADPH donate a hydrogen molecule or a hydrogen atom to it, so that my NADPH turns into NADP, and this little guy has just gained a hydrogen. Right, there you go. So he's gained a hydrogen. This little guy over here, he's also gained a little hydrogen. Okay, so 2 of them have been used. Okay, everyone okay with that? 1 mole of TP becomes RUBP, right? Don't say mole, because that's going to get confusing. ATP is also used here as well. So 2 ATP molecules are used here too. They go in and they make ADP. Okay, that makes my TP. Okay, that's all that goes on. So a bit of hydrogen is added, a bit of phosphate is added. That makes my, let's put a 2 there. That makes my TP/GALP. Okay, final stage, my TP basically has just another bit of ATP added to it. That will go in there. ADP will fall off, right? And my TP can turn back into RUBP. So is H removed from GP to make TP? No, hydrogen isn't removed. It still has the hydrogen attached to it. Here's a little hydrogen attached to it. Here's a little hydrogen attached to it. It just has another bit of phosphate added to it. Okay, so stage 2, add hydrogen, add phosphate, and you turn GP into TP. Okay, final stage, add a bit of phosphate, you make RUBP. Okay. To understand the maths then, if I had 1 cycle of this, so if I had this 1 loop happening, I'm going to do a little 1 here, right? If I have 1 cycle happening, how many GPs am I going to make? So how many GP molecules am I going to make if I have just 1 turn of this? Phosphate is added in both phases at present. Lovely. So after 1 cycle, I have 2 GPs made, as you guys have all seen. Okay, so this 1 5 molecule thing here has reacted with this 1 carbon molecule there, and it has made 2 3 carbon molecules. Out of those GPs, how many TPs are made? Who can tell me that? There's going to be a bit of a gap here. Go right about now. Let's see if that comes up. Lovely. So this also makes 2 TPs. Okay. If I just draw this for you, you can see the predicament that we get ourselves into. So I'm just going to talk about TP from now on. I just wanted you to see that if you've got 2 GPs, that makes 2 TPs. Okay. So a TP looks like this. So 1, 2, 3. And then because we said there's 2 of them, we're going to have that. Okay. If I wanted to turn my TPs into ribulose bisphosphate, which has 5 carbons, look how many I have. So if I have RUBP, so let's have 1, 2, 3, 4, 5. If I wanted to turn that into that, I'd have to use that one, use that one, use that one, use that one, and use that one, right? So I'd be left with just 1 carbon left. Okay, if I did that, if I wanted to make my ribulose bisphosphate with 2 TPs, I'd only have 1 carbon left. Okay, that is problematic because none of these molecules have 1 carbon apart from carbon dioxide, and I can't just turn it into carbon dioxide because there's no oxygen. Okay, if I have, if I did the cycle twice, how many TPs would there be? So if I did this cycle twice, how many TPs would there be? Who can tell me? So if I did this cycle twice, excellent. So if I did the cycle twice, so here's the second cycle. I'm going to have 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Right. So if I do it twice, I'm going to make 4 TPs. Okay. If I try to turn that into ribulose bisphosphate, so RUBP 1, 2, 3, 4, 5. So cross that out. 1, 2, 3, 4, 5. Oh, lovely. I can do that. See if I can do another one. 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. Oh, damn. I'm left with 2 carbons left this time. Okay. None of these have 2 carbons, so I can't do that either. Okay. If I do 3 cycles, this is when the magic happens. If I do it 3 times, I'll make, here's one TP, here's another TP, another one, another one, another one, and another one. Should have 6. 1, 2, 3, 4, 5, 6. Yes, I can count. If I try to turn that into ribulose bisphosphate, 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. Let's make another one. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Let's make another one. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And then muy bien. I have something left. I have something left I can actually use, right? So in this Calvin cycle, in this light independent cycle, and I know this is a bit confusing, if I want to regenerate my RUBP, then I have to do 3 cycles to make 3 ribulose bisphosphate molecules and 1 molecule of TP. Okay, so if I do 3 cycles, then I will make 3 ribulose bisphosphates and 1 hexose sugar. So 3 cycles is really, really important. This means that 1, 2, 3, 4, 5, 6, 5 out of 6 TP molecules, 5 out of 6 TP molecules is used to make ribulose bisphosphate. Okay, 1 out of 6 is used to make a hexose sugar. Okay, so if I have 3 cycles, that is enough to basically make a bit of a hexose sugar. So I need to have 5 out of 6 of my TPs or my GALPs being regenerated into RUBP, and I'll have 1 TP left that can get turned into a hexose sugar. Okay, the only added complication to this, and I know it's already quite complicated, the only complication left is a hexose sugar is made of 6 carbons. And if I only have 1 TP molecule left, 1 TP molecule, look, here it is, only has 3 carbons in it. So to make this process work, as opposed to doing 3 cycles, I actually need to do 6. So if I do 6 cycles, I know this is getting confusing. If I do 6 cycles, I'm going to double the amount of these 2 things. Okay, so I'll double my amount of ribulose bisphosphate, so I'll end up with 6 molecules of that. And I'll double my amount of TP, so I've got 2 of them. And if I've got 2 molecules which have 3 carbons in, I can stick them together and turn them into sugar, right, into glucose. So in this cycle, I think the important fact that you guys need to remember, and I'm going to rub out some of this stuff on the side. So for those of you that want to watch me talk about that again, which I imagine quite a few of you might want to, oh God, this is going to be a nightmare. If I rub out some of this stuff, is this going to work? Oh, you can actually use the little pen as a proper rubber. That's really cool. Sorry, I've just learned something today. If you have a proper rub out over here, and if I manage to write this down in the next like couple of seconds, the key facts that you kind of need to know about this is that you need 6 cycles makes 1 molecule of glucose. Okay, so 6 cycles will make 1 molecule of glucose. In terms of the amount of ATP that I need, each cycle has 2 ATPs here and 1 ATP here. So there's 3 ATPs per cycle. So 3 times 6 is 18. So that uses 18 molecules of ATP. In terms of NADPHs, you just have 2 of them. So that means 2 times 6 equals 12. Do a little S at the end. Okay, so to make 1 molecule of glucose, look how inefficient that is. 1 molecule of glucose requires this whole thing to go around 6 times, which uses 18 ATP molecules, 12 NADPHs, and that will only make 6 ribulose bisphosphates. So I'm going to have a look at your questions, because I'm sure you've got some, and then, in fact, let's put this up. So how confident are you feeling? I'll read some questions as you tell me how confident you're feeling. Let's have a look. Do we need to know NADPH becomes NADP plus. Aliens are coming, watch out. NADP plus is just a way of saying not reduced NADP. Okay, the reason it says it there is just because it's not reduced. Because losing CO2, 6 turns, nice. 2 out of 12 TPs formed a revolve making hexose. 6 turns of cycle produces 18 ATPs, 12 reduced NADPs. Lovely. I'm glad that you said that. So is it not NADPH2? No, because it's a hydrogen atom, not a hydrogen molecule. How does TP result in glucose? Do they join together? Yes, basically they just join together via condensation. Do you need to know these numbers? Kind of. So I've seen exam questions asking you to work out the amount. So what I've always said to my kids at A-level, I probably shouldn't refer to them as kids because they're like 17 and 18, is if you can remember the cycle, so as opposed to just remembering fact after fact after fact, if you can remember RU, BP, GP, TP, right? And you remember that these 3 things turn into one another, right? And if you remember what each one looks like, so 1, 2, 3, 4, 5, 1, 2, 3, I'll do 2 of them, and 1, 2, 3, 1, 2, 3. And you remember that ATP is used here, so 2 of them, and 2 NADPHs, and 1 ATP here, right? If you can remember just that little diagram there, then you don't need to remember any numbers at all. You should be able to work it out in the same way that I've just shown you, right? If you can remember just that, and I would draw that diagram a million times until you remember it, that's exactly what I did, then you don't need to remember any numbers. You can just go through it, and then like if they give you a different question, then apply it. Okay, what else have people said? Lots of 2s in between 2 and 3, that's fine. So that's the equation of photosynthesis is balanced by multiplying everything by 6. I don't think that's why everything is balanced by multiplying the equation by 6. I like to think, Fatima, that is the case, but I think that's more a nice coincidence. Are you going to do exam questions? Indeed. Right, guys, have a go at doing this then. Here is a question related to what we've just been talking about. Okay, it's not too hard. I'm going to give you a couple of seconds just to read it. Or maybe I'll read it out to you. Maybe that would be nice. So the diagram below shows some of the steps in the process of photosynthesis. Okay, so I've got molecule P. That gets turned via the light-dependent reactions into molecule Q. Molecule Q just goes off. Molecule R and S are also made. They feed into the light-independent reactions. Okay, name molecules P and Q, and then tell me what molecules R and S are. So have a think about it. See if you guys can have a go at working this out. See if you can work out what P is, see if you can work out what Q is, and then R and S. Okay, I'll have a quick drink of water. I'm really sorry to disappoint you, Fatima. So let's have a look. So molecule P. So think back to the equation then. So CO2 plus H2O makes glucose, C6H12O6 plus O2. Okay, so light-dependent reactions. What was the light-dependent one? Hopefully you guys remembered that the light-dependent one is that one, and the other light-dependent one is that one. So molecule P, lots of you are saying molecule P is lovely. So molecule P is indeed water. Okay, so water feeds into the light-dependent. Yeah, be careful with that, because some of you are saying like produce NADP, but it's the light-dependent reaction. Those 2 guys do not feed into it; they're just made. Okay. Molecule Q is something which is just basically floating off into wherever, right? So molecule Q is going to be what? If molecule Q isn't really needed, lots of you are saying it, molecule Q therefore has to be oxygen. Okay, so molecule Q isn't needed, so it just floats off through the stomata. Molecule P has to be water. Okay, place a cross in the box next to names of molecules R and S. Look at them trying to be tricksy and give you basically the same thing. Okay, so what molecules did we produce in the light-dependent reaction? What molecule did we produce? Who could tell me? Someone said R and S. Oh, they are R and S. Sorry, that's me misreading this. So R and S. must be. Yep, you guys are saying it. It's got to be ATP, so it's not ADP. Can't be those two. It's got to be ATP, it's not oxidized, and reduced NADP. There you go. It's got to be that bottom one. Okay, so they are the two things that get made in this reaction. Okay, I think that one's not too bad. There you go. Let's just give yourself some marks. Let's have a go at this one. So describe the role. I've got two more exam questions. I've got this one and one last one. So describe the role of Rubisco in the production of GALP. Okay, GALP, if you are not doing Excel, just means TP. So what is the role of Rubisco in the production of GALP in the light independent reaction? Guys, have a think about that. I'll have another sip of water to give you some time to think. And then don't mind me, I'm just going to draw that little diagram I drew earlier. So if I were you, if you've got a pen and paper, I would draw something like this. Okay, so they are asking you to talk about how this is made by an enzyme, which is somewhere over here. Yeah, look how happy that little enzyme is. Okay, I'll give you about 30 more seconds. I'm going to read what you're saying. So if you're saying to catalyze the reaction, you'll never get a mark for that. You need to be so much more specific. So catalyze what reaction? Lovely. Fatima, you are right on the money there. Catalyzers. Don't just say catalyzers. Lovely. So Kitty Payton, you are on 1 mark at the moment if you say just that. Aisha, you are getting 2 marks if you had, in fact, I'm on about 2 marks, that's 4. Aisha, you're getting 4 marks pretty much with what you are saying. Okay, if you can tell me what Rubisco is, then you can get a mark because this question doesn't actually tell you what Rubisco is. So if you can say what it is, that'll get you a mark. If you can talk about that sort of mystery molecule which exists for a few seconds as well, you can get a mark as well. Let's look. So Rubisco catalyzed reaction CO2. Okay, so Nyasha, I think you're pretty good. Rubisco, lovely. Ewan, Rubisco is an enzyme. So let's go through this. So first mark, you guys need to be saying that Rubisco is an enzyme. So Rubisco is an enzyme. That will get you a mark. Nice and straightforward. Lovely. Most abundant enzyme in the world, or as much as we know. Right, so next thing. So lots of people are saying it catalyzes reaction. That is the exact word you need to say. So it catalyzes the reaction. What is the name of that process? I've said it lots of times today. So the process going on right over here. What is its name? It catalyzes what reaction? Who can tell me? Catalyzes the... Lovely, Nyasha. So we want to say it catalyzes the fixation of carbon, or CO2, of CO2 to RUBP. Okay, carbon fixation, spot on. To get the next mark then, let's say what that forms. So when you catalyze that reaction, what is that odd thing that you form? What do we need to say? Who could tell me? Give it a nice generic boring name of exactly what it is. So this forms, not GP. Before we get to GP, Kitty. Yes, lovely. Forming, sorry, lovely, Ewan. Forming an unstable 6-carbon molecule. Probably should say molecule. 6-carbon molecule. That breaks down or is hydrolyzed to form GP. And then final point, which lots of you have said, GP is reduced by NADPH. If I were writing this in an actual exam, I would say Unstable is fine, yeah. I would say reduced NADP. It's broken down, or it's reduced, sorry, by NADPH. And we could say, and is phosphorylated forming TP, okay, or if you are LXL, GALP. Okay, there you go. That is the kind of question that you can expect. Okay, so it doesn't seem too complicated at first, but I think to get full marks on it, you'd have to really think about what you're saying, right? So go through it step by step. So what is Rubisco? Define what it is, enzyme. What it does, catalyzes the fixation of CO2 to RUBP. What does this form? 6 carbon, or an unstable 6 carbon molecule that makes GP. And then finally, because the last step, because it's talking about GALP/TP, you need to save us our steps. So that is reduced by NADPH and phosphorylated forming TP/GALP. Okay, unstable intermediate, yeah, lovely, Wardo, that's lovely. If you don't say unstable, do you still get the mark? I think you probably get the mark. I think with questions like this, the keywords that you need to have said, if you haven't said RUBP, there's no way you're going to get the marks. If you haven't said NADPH, you definitely wouldn't get the mark. Probably want to say ATP down here as well. That'd probably be pretty key to get the mark. They don't mention GP at all either. So it's these keywords here which you're going to have to say to get the marks. Okay, one last question, and then I might save limiting factors, because otherwise I'm going to go even more over than I already am. So one last little exam question. I'm going to skip all that. Here you go. Here is one last little question, and then we'll finish. Okay, so I'll answer your questions for a couple of minutes too. But here is one last little question. In fact, not that question. Sorry, that question. Here's a question I need to have a go at. That one was about something you haven't done yet. Have you done a live chat on respiration? No, that will be later on in the next couple of weeks. Okay, have a go at this question. This one's really tricky, and I think it really throws people off. So in an investigation, single-celled algae were kept in bright light and were supplied with carbon dioxide containing, oh no, I've just had a little panel come down top of my screen, so I can't actually see what it says. Oh, fortunately, I do have it open over here somewhere. Says, was supplied with carbon dioxide containing a radioactive carbon atom. Okay, that means they can basically monitor it. After 300 seconds, the carbon supply was turned off. The graph shows how the concentrations of carbon dioxide, GP, and RUBP have changed. Your camera is covering the question. Oh no, sorry, I didn't realize that you saw that as well. That's pretty stupid of me. There you go. Can you see that thing at the top as well? Let's have a look. I don't know why I'm asking you that, because I can literally just have a look. No. Hopefully you can see it. So the question says, explain why between 0 and 300 seconds, the concentration of radioactive GP remained constant. Okay. So in this graph, look, carbon dioxide is constant right up until 300 seconds. Okay. So in this question, why is it the case? Why is it the case that if I've got lots of carbon, my GP is going to stay absolutely constant? What's going on? Sorry, Aisha, my face hasn't often blocked the question before. I don't know how to deal with that. I have dealt with that. I don't know why I said that. Why is my GP staying constant, right? Why is this guy over here staying constant? Can anyone think of a reason? So if there's loads of carbon dioxide, think about what GP is getting turned into. Okay, so again, I would always draw this out. Okay, if there's loads of carbon dioxide, what is going to make sure that my GP stays constant? Lovely. Yes, lovely. You two, you guys, you've got it. Jaisal and Aisha. Basically, what you need to be saying for this question, because there's so much carbon dioxide, then the reaction between, so I'm gonna word this really basically, so you have to listen to get the full 3 marks. Because there's carbon dioxide, you need to say that CO2 and RUBP are reacting, making GP. So RUBP and CO2 are reacting. Okay, because there's carbon dioxide, my CO2 and RUBP are reacting, forming GP. Then because there's enough carbon dioxide, GP is forming TP, right? And because I've got plenty of TP, TP is going to be regenerated into RUBP. And essentially what you need to say is that GP is being formed at the same rate that it is being, like, I don't know, destroyed kind of thing. So it's kind of in an equilibrium. So you want to say something along the lines of equilibrium will be reached. Okay, because everything is present, GP is turned into TP, it's turned into RUBP, and it's going to stay roughly the same. It might go a tiny bit up and down, but it's going to be mostly the same. Okay, carbon dioxide isn't limiting. You don't really need to say that for this question. We're sort of assuming that it's not limiting because it said it there's so much. In fact, no, actually, no, I disagree with myself. Definitely say that. So lots of CO2 is reacting with RUBP, producing lots of GP, producing lots of TP, producing lots of RUBP, going round and round and round. Okay. Yeah, sorry, converted into gout for you, Aisha. At the same rate, it is being reduced. Yeah, so everything is going at the same rate at this stage. Okay, you haven't even finished AS. Don't worry, Aidan, too much. You've got plenty of time. You don't need to panic or anything. Okay, so last two questions. Explain why between 300 seconds and 380 seconds, the concentration of RUBP increased. So look, my RUBP is massively going up here, and my GP is massively going down. So why would RUBP increase? Who can tell me that? And again, Aisha, if your school's going too fast, don't worry too much. It just gives you time to revise, which is a good thing as well. So why is it the case that my RUBP is going to increase? Yes, Jisel, you're right, but then what isn't happening to that RUBP? So radioactive carbon is used in a reaction of GP, leading to the radioactive carbon in TP, leading to a concave radioactive RUBP being built up. Yes, so Aisha, you're fine. So no carbon dioxide for carbon fixation to occur, hence buildup of RUBP. Lovely. So we need to say something like there is no CO2 or there is less CO2 to react with RUBP. So we can say, let's get this perfect, RUBP not changed into GP, which in turn GP decreases. There you go. So it's basically going to get stuck. So as soon as you take out carbon dioxide, CO2, as soon as you take that out, then basically you have a massive buildup of this. That thing decreases. TP is going to just stay kind of constant. It's going to stay where it is because it's not going to be. In fact, no, TP could get turned into RUBP, so that'll probably go down itself, so long as you've got enough NAD, pH, and enough ATP. Okay, lovely, guys. Yeah, your special math. So go on. I'm going to finish here. I'm not going to do that question. I will do a session on this. Don't you worry too much. You're not going to miss out on anything. Just so you know then, and just so I've done my part to make sure that you know everything that's going on with SnapRevise, I'll show you what we've got. So, let's discard that. If I show you our website, if I can find it, which is here. Guys, just so you know, just if you have been enjoying these like webinars and you are interested in sort of signing up to get more of this, or if you're the lucky person who posts on Instagram, this is basically what you are going to get. So if I click on SnapRevise 2.0, essentially, when this stops being funny, there you go. What we're offering you is basically a way for you to pick any topic you like. We'll test you on it, or you'll get a little test done, which doesn't really involve us very much. You'll then get some really intelligent videos being sent to you. I mean intelligent as in it will work out what you don't know and send you videos and that kind of thing to fill your gaps. You'll then get lots of tests that you can do, literally thousands of tests to build up your knowledge. You'll basically have access to all of our lovely little revision guides. So if you've got a revision guide, I guess it's something like that, but hopefully ours will be more sort of in tune with what you guys need to know. I don't know. You'll have a few more walkthrough videos you can watch. So you'll have people who just go through exam videos with you that you can enjoy. That's not live or anything. That's just on YouTube or will be sent to you via the app. You can do some of our other exam packs that we've got. You'll get some points. And then if you're still stuck, you'll have people like me. So as you guys have basically gone through this, I'll be here to answer your questions at any point. Okay, so that's the website. If you want to carry on, if you are, don't want to look at that one because that one's going to get well confusing. If you have a look at this, if you go onto our SnapRevise sort of account on YouTube, as I said earlier, our upcoming live streams are here. So if you do chemistry and you want to know more about rate, and I don't even know what that word means, but that equation, then my colleague Georgia is doing that tomorrow. So feel free to set a reminder on your laptops, on your computers and phones, so you can watch this. And here are mine for next week. So I'm doing proteins. I don't know where my other biology one's gone. There is another biology one somewhere after that one. I have to go across. Probably there. There you go. So I'm going to be doing some stuff on respiration. Before I do that, I'm going to do something on photosynthesis again to make sure we've covered everything. But for those of you that I was talking to earlier, look, we have so many videos here. If you want to watch someone talk about A-level biology, look, here's reproduction, here's climate change. I think that is something that you possibly may have covered, may not. Biotechnology. Basically, have a troll through these and have a watch of them. Some of them have very, very few views, and I just don't know why. That's so good. So have a look through some of them if you're ever stuck for anything. Otherwise, those of you that have stuck around, I'll show you the code in just 1 second. In terms of SnapRevise 2.0, which is our new service we're offering, there are different sort of packages you can get. This is the really basic one. If you've signed up to SnapRevise, this is what you will already have, right? There is a pro one, which has these cool sort of smart tech quizzes in them. They've got all the exam questions as well. And then if you want our ultimate package, which involves me being the person to talk to you, then look, you can have the ultimate package, which has drop-in tutor and support chat, basically what I've been doing. Okay, those of you that finally wanted that little code to use tonight, here you go. If you want the code to help you with anything, if you want to get a discount, there it is. That'll get you our basic package. And I think if you use this code, you'll also get a discount if you ever want to upgrade, or so I've been told. Okay, access for 2 years in case you are doing a 2-year course, which I imagine all of you are. Okay, hopefully by now then, guys, you should know what the Calvin cycle is. The light independent reactions. Oh no, I left my objectives on, damn. Limiting factors, we didn't get there, did we? So that will be the next session. This will also be the next session too. Okay, if anyone has any questions for me, feel free to ask them now and I'll try and respond to as many of them as I can. Okay, in terms of physics, at the moment, I don't think so, but I think coming quite soon, we will have a physicist with us. Any questions about this at all? Oh, and I don't know who won free account just yet, for whoever's asking that. You possibly might get told at some point in the near future. I don't know. Any questions? I'm happy to talk about any of, where's it gone? Any of this stuff if you want me to. No? Okay, in that case then, guys, let's discard that again. I am going to go. So thank you so much for coming and watching this. Hopefully it's been a really good use of your time and you haven't felt that this has like made it more confusing. Yeah, come back. Watch a few more videos, watch a few more things. Hopefully this will be really straightforward. Hopefully you guys are doing photosynthesis at the moment and you can go and impress your teacher by showing them everything that you know. Okay. Have a great evening, guys. See you later.